This is Cliff Bueno de Mesquita here, giving a presentation for the BDEE conference. I'll be talking today about microbial drivers of methane emissions from unrestored industrial salt ponds. This is work that was done in collaboration with Susanna Tringe at the JGI and her lab group members, Jingli, Susanna, Wyatt, and Yi. So thank you for attending my talk. I'll be talking today about wetlands, which most of us already know are really important ecosystems globally. They store vast amounts of carbon, they're important habitat for numerous wildlife species, and they perform important ecosystem services for humans, such as flood control. Unfortunately, many wetlands have been degraded over the past couple centuries due to human activities, and development has converted them to other uses. One such use is for industrial salt production, and this is particularly important when we're talking about coastal wetlands near the oceans that have been evaporated for industrial salt production. The good news is that more recently, there have been more efforts to restore wetlands back to their former states, and that's what I'll be talking about today, with a case study from some wetlands in the San Francisco Bay. So our study site is in the South San Francisco Bay. We sampled 2 unrestored salterns. So saltern is just, I'll be using that word to refer to an industrial salt pond for industrial salt production. There's 1 restored saltern, so it used to be a industrial saltern and it's been recently restored, and then 1 unaltered, what we call historic wetland, so it was never used for industrial salt production. And the unrestored salterns are no longer in use for industrial salt production, but they have not been restored, and so you'll see how that plays out in the results. We took 3 sediment cores at each site, and they were split into 2 depth profiles, the 0 to 5 centimeter and the 5 to 15 centimeter. So that's 6 samples from 4 sites, so a total of 24 samples. We then did abiotic characterization of the sediments for carbon, nitrogen, phosphorus, salinity, dissolved oxygen, several different cations such as potassium and magnesium, and several different anions such as nitrate and sulfate. We used DNA sequencing, including 16S marker gene sequencing and shotgun metagenomics, to characterize the taxonomic and functional profiles of the microbial communities in these sediments. We also collaborated with IMSL, the Environmental Molecular Sciences Laboratory, to do some metabolomic work on these samples. And we performed metabolic modeling using the KBase online tool, which was developed by JGI. And this was done using metagenome assembled genomes. So these were genomes assembled from the shotgun metagenomic data. And then lastly, we did a comparative survey using Joint Genome Institute's IMG database. It's another online database curated by Joint Genome Institute, and many researchers submit their metagenomic samples to this database, so we were able to look at a wide variety of samples. First key question we wanted to answer is, how did industrial salt production alter ecosystem structure and function? And particularly in terms of function, we wanted to look at the impact on greenhouse gas fluxes, in particular carbon dioxide and methane. And since there are current restoration measures going on in the San Francisco Bay Area and elsewhere, we're asking, are these restoration measures effective, particularly in terms of the microbial community and the greenhouse gas emissions? So typically the restoration measures involve reestablishing the hydrologic connectivity of the wetlands, So we want to see how is this affecting the microbial communities and greenhouse gas fluxes. So getting right to the results, I'll first go through the abiotic differences among the sites, as well as the methane emissions. So in the top panel here, we have some of the key chemical variables, and we see in all four cases for salinity, sulfate, inorganic nitrogen to phosphorus ratio, and the methane flux, in all these cases we see significantly higher levels in the unrestored salterns compared to the historic wetland and the restored saltern. Now on the bottom four panels, our y-axis is now methane production, and we're looking at correlations of these variables with methane production. And note the log axis scale here, so we're going quickly from 1 to 1,000. Methane production in the historic wetland is basically 0 or even slightly negative. We've got very low production, around 10 micromoles per meter squared per day in the restored saltern, and then much more elevated emissions, closer to 1,000 in all the unrestored saltern samples. And these are positively correlated with salinity, sulfate, nitrogen to phosphorus ratio, and negatively correlated with carbon to nitrogen ratio. And if we look at even more variables, we see similar trends. We've got higher levels of a number of different chemicals such as calcium and magnesium, potassium, and these are all associated with the higher methane production in the unrestored salt ponds. Interestingly, we also have a couple negative associations down here with copper, Olsen phosphorus, dissolved oxygen, and iron. And so this is an interesting one. Lower oxygen levels being associated with higher methane emissions makes sense. Most of the methane production here is likely anaerobic in these sediments. Now moving on to the microbial community, we see that there's actually similar diversity, and this is just the Shannon index in the historic and the restored, and then much lower diversity in the unrestored samples. If we take a broad look at the taxonomy at the phylum level, we see higher Euryarchaeota, Bacteroidetes, Firmicutes, and alpha proteobacteria in the unrestored salt ponds. And we have higher delta proteobacteria and chloroflexi over here in the historic and restored wetlands. In terms of the beta diversity, we have a PCOA plot here, and this is showing some clear community separation between our three sample types. Interestingly, the restored and historic samples are also significantly different from each other, but they're not as different as they are from the unrestored salt ponds, which is differentiated along axis 1 here. And then lastly, we have a little bit of a functional profile here where we assign some of the known taxa that are involved in iron and sulfur, nitrogen, and methane cycling. And an interesting thing here is we actually have a lot more relative abundance of sulfur reducing bacteria in the restored samples and even historic samples compared to the unrestored samples. And they're a very small proportion of the community, but you can see here in the unrestored samples we actually have some taxa that are known to be associated with the methane cycle. Now, using the shotgun metagenomic data, we assigned genes to these KEGG orthology groups here, and so this is just a heat map of some of the genes involved in carbon, nitrogen, phosphorus, and sulfur cycling. And I've just highlighted 3 groups here where there are clear differences between the unrestored and the historic and restored wetlands. So there's actually higher abundances of methane and ammonia oxidizing genes in the historic and restored samples, and there's higher methylphosphonate cycling genes. So this is the PHN group over here. Higher all across the board except for this one sample. And then importantly, for these methane cycling genes down here, the MTR group and the MCR group tends to be much higher in the unrestored samples compared to the restored and historic samples. In terms of the metabolic differences, there were a couple compounds that came out as different. Unfortunately, we don't have data yet for the restored samples. So this is just the Saltern versus the historic. And we see higher levels of dimethylsulfone and trihalose. And so one thing this is sort of suggesting is that there are these differences in sulfur cycling and carbon cycling, and perhaps differences in the types of organic matter and organic carbon sources that are being cycled in these different wetland types. Okay, so now let's move on into looking at really what is driving methane production. So these three plots correspond to three different ideas. So the first is the hydrogenotrophic archaea. So this is the MCRA gene involved in classic archaeal methanogenesis. It's positively correlated with methane production. We have the PHNJ gene, which is involved in methylphosphonate cycling, which has been shown to produce methane during the cleavage of the carbon-phosphorus bond. This is performed by bacteria. And then and this is also positively correlated with methane. And interestingly, we have methane and ammonia oxidation, PMO and AMO-A genes. And so these are actually negatively correlated with methane, and this makes sense because these organisms are actually oxidizing methane. In other words, they're consuming methane. So to do a follow-up experiment, we did a substrate incubation experiment, which ended up showing some evidence for methylotrophic production of methane. And so we used unrestored saltern sediment from one of the unrestored salterns in an anaerobic chamber that we spiked with trimethylamine, methanol, acetate, and then control. And you can see that the trimethylamine and the methanol spiked samples ended up producing the most methane over the course of 14 days. So, which microbial taxa are involved in methanogenesis in these samples? So we ran the metabolic models using the MAGs, the metagenome assembled genomes, to get different reactions that are occurring in the samples. And then we pulled out reactions from these 4 KEGG modules. So there's 4 KEGG modules involved in methanogenesis. There's the methanol pathway, the acetate pathway, the methylamine pathway, and the carbon dioxide or hydrogenotrophic pathway. And so we were able to identify the MAGs that were associated with reactions in these modules. And so we identified 13 genera that were performing these reactions. And the key one is this Methanolobus archaea. This is the one that's actually performing the final step of the methane production. And then these other ones are performing some of the intermediate steps. And you can see that 3 of these genera are known halophiles. So these were likely selected for during the course of the industrial salt production, which vastly increased the salinity in those samples. And the salinity is still high in these samples. But these 3 halophiles are involved in some of the intermediate steps in methane production. So, lastly, we wanted to look at how widespread methane production in saline environments could be. So we did a survey of metagenomes available on the IMG database, 736 metagenomes. And we see that the key taxa are most abundant in our unrestored Saltern samples, but they're also present in other samples. So here is our unrestored Saltern over here. This is relative abundances in terms of percent. So actually, across the board, these are pretty low percentage abundances. However, that doesn't mean that they can't be important for the greenhouse gas cycling. And you can see down here, there's just a number of other samples. These all have in common are that they are at least the salinity of the ocean, and in some cases, they're even more saline or hypersaline. And they include deep ocean vents, hydrothermal vents, methane seeps, hot springs, estuaries, coastal wetlands, salt marshes, mangroves, ocean, hypersaline lakes, lagoons, coastal ocean samples. And these are all sediment samples. So these key 13 genera tend to be slightly more abundant in our unrestored salterns, but they're actually present in most of the other samples. Now, if we look at the same data set and we pull out the MCRA methanogenesis gene, we see that our unrestored saltern is actually not too special. It has somewhat elevated levels, but not as high as some of these other sample types. Particularly certain ocean sediment samples have much higher levels of MCRA. And these are, in terms of analysis, these were normalized with the DSeq-R package. And then lastly, if we look at the pH and J gene in all of these samples, we actually do see that our unrestored saltern stands out in terms of having the highest abundance of pH and J. However, there are also some other samples that have high levels of pH and J. It's just not across the board in these sample types, but certain locations in the ocean or in the methane seeps or in coastal wetlands. Some samples have high levels of pH and J. And this could be related to phosphate levels. Typically, there's greater levels of pH and J when phosphate is limiting. So in conclusion, we see elevated methane emissions from former industrial salterns. This is associated with high salinity and sulfate levels. And it looks like there's a combination of hydrogenotrophic and methyllotrophic methanogenesis, including methylphosphonate cycling. The good news is that restoration can help mitigate. So by taking restoration measures such as, most importantly, restoring the historic hydrology and restoring hydrologic connectivity to these former salterns can change the abiotic conditions and microbial communities and actually have an impact on the greenhouse gas emissions. Interestingly, when we compare these industrial salt ponds to other diverse saline environments, some of these other environments also have similar MCRA and pH and J levels, and the key methane cycling taxa present. So the interesting follow-up work would be to perform more methane measurements in some of these other samples. And there's already been some work on measuring methane in ocean samples coming out of the water column. And so it would be great to build on this work in some of these in the open ocean, as well as some of these other environments like hypersaline lakes and other coastal wetland areas. So we'd like to thank the U.S. Department of Energy and Early Career Research Program for funding, also the JGI sequencing staff for all their help. I'd like to acknowledge my advisor, Susanna Tringe, and the rest of the Microbial Systems Group, past and present members. And we'd like to thank John, Donna, and John for access to the field sites. And thank you for looking at the talk, and I hope I can answer any of your questions over the course of this virtual online pandemic conference. So thank you.